Now inviting OP260 to present.
Good evening, everyone. Myself is Dr. Gomati Swaminathan. My title of the presentation is Central Neuroproductive Effect of Cyclopeltata on Fluoride-Induced Neurodegeneration in ST rats. Next, madam. Next, about introduction. The groundwater is the most important source for various purposes in many parts of the world. People are using the groundwater for drinking and other domestic household purposes like cooking and so on. Nowadays, nearly 29 nations of the world and 19 states of India, India's groundwater is floated with fluorinated fluorine. It may be the fluorine is less than 1 milligram per liter means it is healthy, beneficial for health. It may be it crosses more than greater than 1.5 milligram per liter means it makes many health hazards and health defects. It affects many parts of the body like brain, kidney, heart, and so on. So that's why we selected the fluoride-induced neurotoxicity for our studies. And here we went for herbal medicine. We selected herbal plant that is the Cyclopeltata lamb, belonging to the family is many Thermaceae family. So this plant was investigated and reported for many biological activities like nephroproductivity, antidiuretic, antidiabetic, anti-ulcer, and so on. Many work had been done on this plant, but except these neurodegenerative diseases. That's why we taken the plant for studies. And one more point is this plant is having potent antioxidant content, phytoconstituents like alkaloids, especially benzoisoquinoline alkaloids are present in this plant. That's why we selected the plant for further investigation. In Tamil Nadu also, many parts of Tamil Nadu affected because of this fluoride contamination, especially in Dharmaburi and Krishnagiri and Salem district and nearby Namakkal. Namakkal also, Vennanthur, Coimbatore, Satyamangalam, every parts of nearby Namakkal district and this Salem district, most of the groundwater is floated with fluoride. That's why we selected this fluoride-induced neurotoxicity for our study. This is the environmental toxin which will enter to our body means automatically which creates the pro-inflammatory cytokines and which leads to produce the reactive oxygen species. So oxidative stress will form and finally will get the neuronal death. So it would be retarded by the aid of antioxidant pre-treatment from nature. We selected the roots of the plant for further studies. Aim and objective. The aim of the present research work is to investigate the preliminary phytochemical and neuroproductive evaluation of ethanol extract of Cyclopeltata roots. And objectives are: study was designed to investigate the following. First one is the preliminary phytochemical study and in vitro free radical scavenging activities and acute oral toxicity studies and neuroproductive activities. The injection method is fluoride-induced neurotoxicity and behavioral studies for anxiety and depression and biochemical estimation, in vivo, brain, thermal, enzymatic analysis and extend analysis and histopathological studies and statistical analysis. Materials and methods. The plant was collected from Trivandrum district, Kerala and authenticated by Dr. B. Duraiswamy, Head and Professor, Department of Pharmacognosy, JSS College of Pharmacy, UP. This is the authentication number. And the plant material was successively extracted with petroleum ether, chloroform, ethyl acetate, and 90% volume by volume of ethanol in oxidate apparatus. And all the extracts will be subjected to preliminary phytochemical study and in vitro free radical scavenging activities. And part 2 is pharmacological studies. First of all, the protocol was got approval from Institutional Animal Ethical Committee. This is the reference number. And acute toxicity studies was done by OECD guidelines 423. Here, up to the dose level 2000 milligram per kg, there is no mortality. That's why we selected 110th and 120th dose. 100 milligram per kg low dose and high dose is 200 milligram per kg. Then animal grouping. The group will be divided into 5 groups. The group 1 is the normal group, control group, and group 2 is disease control. fluoride 120 ppm for 30 days. And the group 3 is standard group, indomethacin 0.5 milligram per kg along with fluoride for 30 days. And the group 4 and group 5 is ethanol extract of cyclopeldata 100 and 200 milligram per kg along with fluoride infused drops. First, we evaluated the 7th day and the 14th day. We evaluated the all the days we evaluated the general behavioral studies such as food intake, water intake, body weight, and all. But behavioral test for anxiety and depression evaluated at the 7th and the 21st day. We done locomotor activity and swim test for anxiety and depression. And animal were sacrificed at the 30th day. And finally, the brain is isolated and will be subjected to in vivo enzymatic antioxidant enzymes like superoxide dismutase, catalase, glutathione peroxidase, and glutathione reductase. And result and discussion. The part 1 is phytochemical studies. The phytochemical study states that this plant contains alkaloids, flavonoids, phenolic content, tannins, saponins, carbohydrates, trisulfinoids in these all the extracts. And here, on the basis of preliminary phytochemical studies and in vitro free radical scavenging activity studies results, we selected the total, we estimated total phenolic content and total flavonoidal content. Initially, we got phenolic content is 46.34 milligram per gram and flavonoid content is 48.12 milligram per gram. Then, in this in vitro free radical scavenging activity, we compared the IC50 value with the standard. In this PPPK radical scavenging activity, nitric oxide radical scavenging activity, in reducing power, this ethanol extract shows significant IC50 value. That's why we selected the ECP for further pharmacological studies. In general behavior, the food intake, water intake, and body weight is decreased because due to the fluoride-induced neurotoxicity. So, which was recovered by ethanol extract. Here, the next one is the behavioral test for anxiety and depression. The first one is locomotor activity. Here, the number of movements in animals in 5 minutes was decreased in fluoride-induced rat when compared to control and standard drug. Then it was recovered by ethanol extract of 100 and ethanol extract of cyclopeldata 100 and 200 milligram per kg. Here, the number of movement is decreased, which indicates the presence of depression in the animal, anxiety and depression in the animal. It was recovered by this extract. And next one is the enforced swim test also. The immobility time of the fluoride-induced rats shows the presence of depression. Because of depression only, the immobility time is more. So then it was recovered by ethanol extract of cyclopeldata root treated rats. Next one is biochemical analysis. Here, the fluctuated level, the SOD and CAD and glutathione reductase and peroxide level is decreased in fluoride treated rats, which indicates the antioxidant response mechanism is fluctuated because of fluoride treatment. It was recovered by ethanol extract of cyclopeldata. This is the histopathological studies. This is the CA1 hippocampal region of the brain. When compared to control, the fluoride, the degeneration is more. Okay, which indicates the presence of neuronal damage. So, this is recovered by ethanol extract of cyclopeldata 100 and 200 as like control and indomethacin. Conclusion. From our studies, it can be concluded that ethanol extract of cyclopeldata is non-toxic and safe, and it possesses significant neuroproductive activity against fluoride-induced neurodegeneration in the tested dose level 100 and 200 milligram per kg, and also possesses antioxidant activity and promotes the enzymatic and antioxidant defense system in neurodegenerative conditions induced by fluoride. The potential neuroprevention might be due to the presence of phytoconstituents such as alkaloids, flavonoids, and coumarin in this plant. So these are the references we followed. Thank you so much.
Congratulations, it's a very good work.
Thank you, sir.
Among the various plans that are used for such studies against fluoride, why did you select this particular plan? What is the rationale for selection?
First of all, I saw the literature, sir. Many work had been done on this plant. For example, nephroprotective, like that, many work except these neurodegenerative diseases. That's why that is the first reason, sir. I selected the plant. And the next thing is it contains potent antioxidants also, some alkaloidal substances. Okay, and one more thing, I decided whether the plant is having any other active constituents or not. That's why I selected the plant for the studies, and very rare plant also, especially it is available in Tirunelveli districts and nearby Kerala. So that's why I selected the plants.
Why did you take the route? What is the rationale?
Even for root also, sir, already some literature reported the root is having benzisophenolene alkaloids. Sir. For example, already they isolated some perforin-like compounds. Sir. That's why I decided to select the root.
Okay, then why ethanolic extract?
Most of the active constituents are soluble in ethanol, sir. That's why, sir.
Okay, what were all the good collection practices that you followed when you took the route?
First, we collected the plant, sir, and we cleaned the plant and washed.
Collection practices, that means young root, old root. In which season did you collect? Where did you collect?
Okay. Now I got it, sir. The month of April and June, we collected the plant, sir.
Why?
The month of that is only the season. There is 2 seasons for season. We will get the plant more in the month of April and June and November and January, sir. So during the month of April and June, I was collected.
Okay, how did you arrive at the dose of 100 mg per kg?
On the basis of that acute oral toxicity studies, sir. 1/10 and 1/20th dose we selected.
Okay.
That is 200, 1/10. The 10th, 20th is 100. And the IEC also, they never give more animals for studies. That's why we must restricted the groups also.
Okay. Good work. Over to my colleagues.
Thank you so much, sir.
Yeah, Mr. Gumti, is good work. A couple of questions. Is there any other variant of this plant?
Yes, sir. Some of the plants also there, sir. Sir?
What are those? Is there any other variant of this plant? And what are those?
Some of the plants are that same species, sir. Same family, and some other species also available, sir. Certainly, I'm not getting the.
Achha. So, have you cross-checked that this kind of work is done, particularly into MAPA and those kind of abstracts?
Okay, sir. Already there's a cyclia in well data. Some difference is there, sir. Some same family, some other plants also reported, sir. I will check it, sir.
I'm asking you, have you checked the database like MAPA?
In some natural databases, we checked like coconut and that impact and some Chinese databases and Zing databases. These and all we checked.
And this work is not reported anywhere?
Yes, sir. This is not reported.
Achha, what is your future plan about this one?
Next plan is now we are this is the part of work, sir. This is the part of work, and we are going for next plan is isolation. With isolated compound, we will do some work along with the some computer aided drug designing.
What sort of computer aided drug designing are you talking about? What are you going to do with that?
First, then now we will go for some isolation. Then we will see some lead compounds. Okay, from that we will go for some semi-synthetic pathway. That one also we will check molecular talking. As per
What target you are planning for molecular tracking?
P2X7 target.
Is it quite validated target?
Alzheimer.
No, is it validated target?
ATP-gated ion-gated channel receptor.
Okay, all the best. Thank you.
Thank you, sir.
Yeah, I have two questions to you.
Sir,
So this is you taken some geographical information, right? For India.
sir?
Your work based on some geographical related information, right? Because the fluoride contents
Yes, sir. Yes, sir.
spread in means available in some area of Tamil Nadu and
Tamil Nadu, Dharmaburi district, that Krishnagiri, Salem, and nearby Namakkal. Especially we selected, sir, Namakkal district and Salem district. That's why only we selected, sir, because the fluorinated water is more. That's why we selected.
what extent? What extent?
More than 1.5 milligram per liter, sir. That's why we selected. As per
what is the recommended limit?
Less than 1 milligram per liter recommendable, sir.
Recommended. Okay. So are you carried out any geographical informative system used softwares for your studies?
No, sir. No, sir. Thorough literature only we done. Especially we collected some information from Erode district and Salem district and that Dharmaburi district related literature we done. And when compared to all other district, it contains more fluorine contamination. That's why we selected.
You are correct actually, but my question is, when you are going for this geographical work, if you go through for a GSI softwares are there, so where
Okay,
you can accurate information about the locality and in and around for India and in worldwide, and even that particular software provides the accurate information regarding from the content and not only for the fluoride and other minerals. Okay, anyway, you are targeting for the fluoride.
Sure, sir. I will perform, sir. I will take these words and I will implement.
I recommend you have to incorporate in future course of studies, especially whenever going for this kind of studies. If you have the assistance of that particular software, it is more useful for you, and it is in the worldwide because you are not going to be speaking only with the Tamil Nadu.
Okay, sir.
Okay. So I am right now. I'm in Telangana, in Hyderabad. Here, district is called as Nalgonda.
Yes, sir.
Nalgonda have complete fluoride concentrations are more over here.
Yes, sir. Especially in Rajasthan and that Andhra Pradesh, Telangana, everywhere fluorinated water is more.
Exactly. So
okay, sir. Sure. I will do it. Sure, sir. Further studies, if before going further studies, I will do it, sir. Sure, sir.
Okay.
I will follow, sir.
What are the active constituents are there?
Actually, in this extract only we did, sir. Next, now we are in isolation, but already they reported perperin and many other alkalides also, sir. For example, perperamine and tetranthrine and cyclidrine, like that they reported.
So you only done for the extraction, not for an isolation.
Now, sir, now in the spectral isolation over, sir, now it's under spectral characterization.
Okay, what kind of spectral you given for the samples?
We given the sample for IR, NMR, H1 NMR, C13 NMR, and mass spectroscopy, sir.
Okay, what is your expected result? Because based on your literature.
Based on literature, the presence of active constituents, we will get very good. Already we got very good significant activity against neurodegenerative diseases. If maybe we targeted alkaloids and some flavonoids, if maybe we get means next we will go with that isolated compounds. But before that, I will refer
Yes,
that geographical.
catch my question.
Hmm.
So based on your existing literature, what are the active constants are present? Can you list out?
Based on the literature, what are all the active constituents present? Okay, sir.
Yes, that's my question. That is a question.
Cyclitrine and perpramine and perperin and its derivatives, especially Benz isoquinoline alkaloid and derivatives, and already reported detrentine.
So right now you are not having any spectral data with you.
We given the sample. Given the sample, sir. We are waiting for the result.
So not at okay, not at your body.
We given the sample to IIT.
Okay. Okay. And regarding one more question about the GAT studies, you said that for the future plan, you are going for a
Yes, sir. Yes, sir. Yes, sir.
So, what is your plan? Okay, your target, sir, asked you told about for the targets. Right, Alzheimer's.
P2X7 receptor target.
Okay, so this is the only single target it has. This plan is going to work it out, or any other further some other targets are there?
Some other target also are the acetylcholinesterase and a few butylcholinesterase target also, sir. But I was selected that P2X7 might I selected single target first.
Okay, anyway, you have
because sir.
First of all, you have to isolate, right? The isolation.
Oh yes, sir. Yes, sir. Hello, sir.
Yeah. In docking studies, I think you have some idea, right? Whenever you're planning, you have some idea about the gas.
Yes, sir. I have some idea, sir. Along with my HO. Sir, I am not getting, sir. Voice is not audible. That's why.
What kind of docking studies you are going to be planning? Means your target is ready. Means your target is already said, right?
Yes, sir.
That
yes,
particular which compound is active among your
sir. Among my
your target you fixed already.
Yes, sir. I fixed a target.
Yes. So what are the active constituents going to be target means lead? I'm asking about lead. Which lead is going to be target this particular enzymes?
I am going to target that isoquinoline alkaloid, sir.
Isoquinoline. Why?
Isoquinoline already they reported few compounds of Benz isoquinoline alkaloids. That's why I am going to target as per my spectral data result. I will check it thoroughly. Then I will go for I will select the lead compound from alkaloids.
So can you give for example for isoquinoline containing alkaloids?
Perperin.
So they have some already proved one.
Yes, sir. But my spectral data is not done, sir. If maybe I will get any new derivatives, means I will expect, sir.
Okay, thank you.
Thank you so much, sir. Thank you, sir.
Yeah. How many more participants are there?
I think it's over. Chavi, it's over.
And
Yes,
now
sir.
I would
Okay,
like
yeah.
yeah. Yes, sir.
You're done with presentation, sir.
Yeah. Yes,
You are
sir.
done.
We are
Okay.
done. So I would like to invite Dr.
Let's continue.
Kalirajan for the voting.
Thank you, all the evaluators. So kindly give your marks, finalize your marks, and send by mail. So over to Dr. Vikram to give the conclusion remarks.
Mr. Kalirajan. Kalirajan.
Yes, sir.
Thank you, Gopal sir, Principal, Government Pharmacy College, Pondicherry, and thank you to Dr. Rakesh Somani, Principal, School of Pharmacy, Dr. D.Y. Patil, University, Navi Mumbai, and thank you, Dr. R. Sudhakaran, Principal, Vijaya College of Pharmacy, Telangana. for your precious time to evaluate our oral presentations in PharmaNest 21. Thank you, everyone, and a special thank to all the presenters who have nicely explained their valuable research work.
Congratulations to OPF, and thanks to my colleagues, Dr. Somani, Dr. Sudhakaran sir, and very happy to be with Dr. Kalirajan sir, and thanks to the what you call anchors. We had two nice anchors who guided us through the entire program. Thank you all. Thank you one and all.
Thank you, sir.
Stay safe.
Thank you so much, sir.
And congratulations to the participants. You all have done a great job. All the very best. God bless you.
Nice meeting you, Dr. Gopal sir.
Thank you, sir. Pleasure being with you, sir.
Good to hear your questions also, and it was interactive one.
So thank you to providing me an opportunity, as well as I congratulations for all the participants. And thank you, one and all. And please be safe. So I think again at the be safe.
Thank you.
Thank you.
Thank you, sir.
Thank you all,
Thank you,
Mr.
Sudhakaran
Vikram
sir.
and
Namaskar.
Sri. And Namaskaram, sir. Namaskar.
Vikramji, thank you.
Thank you, sir.
Congratulations, Vikramji.